Good morning, grade 11 students. This is lesson number 2, part 1, about prokaryotes. Please make sure to open your books on page 580. The objectives of this part of the section is to identify prokaryotes classification and determine the structure and function of them. Prokaryotes are unicellular organisms, that is, they consist of a single cell. They have a DNA that is located in the cytoplasm and not in the nucleus. Biologists have classified prokaryotes into bacteria and archaea. Bacteria live almost everywhere. They live in salt and fresh water, on land and within the bodies of humans. Bacteria is usually surrounded by a cell wall that protects it from injury and determines its shape. The cell wall of bacteria contains peptidoglycan, a polymer of sugar and amino acids that surrounds the cell membrane. Some bacteria, such as the E. coli, have a second membrane outside the peptidoglycan to make them more resistant to damage. In the following figure, you will see the cell wall in light orange, which is made up of peptidoglycan. The inner wall is the plasma membrane and is in darker orange, and the outside membrane, the capsule, in blue color, which makes the bacteria resistant to damage. Some prokaryotes have flagella that they use for movement. Others have pili, as in the bacteria E. coli, that serve as an anchor or it sticks to a surface or to another bacteria. Let's move on to discuss archaea. But first, we're going to compare between bacteria and archaea. Both bacteria and archaea are small. They lack a nucleus and have a cell wall. However, the difference is that archaea lack a peptidoglycan in their cell walls and their membrane contain different lipids. Archaea live in extremely harsh environments, example, living in environments with little or no oxygen, such as thick mud or the digestive tract of animals. Other archaea live in extremely salt environments, such as the Great Salt Lake of Utah or the Dead Sea, or in hot springs where temperature approach the boiling point of water. Structure and function of prokaryotes. We will explain how prokaryotes vary in size, shape, the way they move, and in the way they obtain and release energy. The size of prokaryotes range from 1 to 5 micrometers, making them smaller than human cells. They have 3 different types of shapes. Rod shapes are called bacilli, spherical shapes are called cocci, and spiral shapes are called spirilla. How do prokaryotes move? Some prokaryotes do not move at all. Others can propel by their tail, the flagella. Some glide slowly along a layer slime-like material they secrete. How do prokaryotes make energy and spend it? Prokaryotes need a supply of chemical energy which they store in the form of sugar. Energy is released during respiration or fermentation or both. In the previous sections, we have discussed what are autotrophs and heterotrophs. Heterotrophs are organisms that rely on other organisms for energy, while autotrophs are organisms that can produce their own food using light or chemical energy. Prokaryotes can be heterotrophs, that is, feeding from the environment or other organisms for energy, can be photoheterotrophs, like basic heterotrophs but also use light energy. They can be photoautotrophs, that means use only light to make energy, and chemoautotrophs, that means use chemical reactions to produce energy. End of lesson 2, part 1, section 20.2. Thank you for your attention.